Okay, so welcome to another in the series of bite-sized bioinformatics, little sort of short seminar style presentations on bioinformatics topics. I'm Simon, and today I'm going to talk to you about something that we've developed relatively recently, but we've kind of been doing informally for a bit longer than this. So we're going to talk about an R package called Anchorview, which sits on top of the Seurat infrastructure and can be useful to help visualize the way that data is going to be integrated. So I'll do a little bit of theory and show some slides to start with to sort of set the scene for this. And then we'll do everybody's favorite thing, which is a live demo and see if we can actually make it work and show you what it does. So if we start from this. So the problem that Anchorview is there to solve is, or to at least help with, is the issue of data integration in single-cell data sets. Mostly we're talking about single-cell RNA-seq here. And this is a common problem in any experimental setup where you have more than one library in your experimental design. So very commonly, after you've run the first part of a single-cell data analysis, where you bring in the data, you do some filtering of it, you do your dimensionality reduction to get a t-SNE or a UMAP plot or something like that, and then you plot out the spread of cells that you get from that, you see something like this. So here we have the cells from two different libraries which have been merged together in the data that we're looking at here, and they've been colored separately on here. So we have some control cells and some stimulated cells, and what you can see is that these cells basically never sit on top of each other. So when we're looking at this data, normally our workflow would be to try and identify cells of equivalent cell type, and we would do that across all of the different samples that we were looking at. And in order to do that, we would want them to kind of sit on top of each other in a sort of t-SNE or UMAP spread. If we look at it on here, we can see that there is no overlap really between the blue and the red. But if we look, we can pretty much see where the structure should be. UMAP and t-SNE don't know anything about the biology that we're chasing. All they're doing is looking at ways in which they can use the signal in single-cell data to separate out our cells. And it doesn't know whether the signals that it's chasing are biological signals to differentiate between cell types, or technical signals like batch effects, or possibly real biological changes that happen as a result of stimulation in this case. If we look at this data, we can see that there is some sort of obvious structure. So over here, you can kind of see this sort of shape here mirrors that, and these mirror that. So if you sort of looked at it for a little while, you could probably pick out that these cells in my controls were probably equivalent to those cells in my stimulated. These cells in the control will probably be the same as these cells in the stimulated. And you can sort of manually see that in some cases. This one's a particularly nice example. But for doing our analysis, we're going to need a more sort of structured and formal way of doing that to bring those cells together. The process that is used within the standard sort of Seurat workflow is, if we take a very schematic view of taking two data sets that we've clustered out with our sort of dimensionality reduction spreads, and which here I've colored by the cell type that the different subgroups have, we can see that although we separate out the cell types in each sample, they don't necessarily appear in quite the same place between samples because of the batch effects or biological differences between them. The integration process that is used in Seurat defines what are called anchor points, integration anchors, which are really just identifying points of commonality between the two data sets. So at a very large level, it's saying, well, this cell cluster over here is going to be equivalent to that one, this one is equivalent to that one, and so on. So it's identifying places which are equivalent in the two data sets, and then it uses those sort of anchor points to be able to then skew quantitatively the data underneath the plot that you're looking at, so that when the integration is, or when the dimensionality reduction is run again, then the effects between the samples are removed and the equivalent cell types should sit on top of each other. How are these anchor points actually defined? Well, getting obviously a correct set of anchor points is crucial to being able to correctly integrate your data. The main method for defining integration anchors in this sort of data is the concept of mutual nearest neighbors. So what's happening in this case is that for every cell in this data set, you take that cell and you use some metric to find the closest cells to that cell in the other data set. So you do some sort of measure of similarity for the transcriptomes of every cell in this data set to every cell in the other data set, and you pick a small number of the closest cells, the closest neighbors between data sets. So here we can say, well, if we take every cell in data set 1 and we find the 3 closest neighbors in data set 2, we could pick some cells and see what they would do. So this blue one would find sort of 3 blues over here, the red would find the green would find 3 greens over here, and mostly they're going to pick out cells of the equivalent type. After we've done that and taken every cell in dataset 1 and found the nearest neighbors in 2, we just do the same thing the other way round. So we start with all the cells in dataset 2 and we find all the cells in dataset 1. Okay. Now, in some cases, this is going to be nice and straightforward because the blue cells over here are going to find other blue cells over here, and the greens are going to find greens over here. But in this instance, I've shown some examples where there is a cell type in my second data set which isn't present in my first data set. So when you have circumstances like this, it's obviously important that you don't misidentify that cell type or try and integrate it to an incorrect set of cells over here. Okay, we want that to be left on its own and not sort of brought into one of the other clusters. Because we've done the nearest neighbors both ways round, what we then find are the mutual nearest neighbors. So we find pairs of cells who are each other's nearest neighbors within some small number of neighbors, and depending on how close your data is, you might need to sort of expand the number of nearest neighbors. You can play around with some settings from that. But by bringing together all of the pairwise comparisons and then finding the ones where they are mutually nearest neighbors, you can identify the common integration points between the data sets and use those to then sort of quantitatively align the underlying data. Now, this is all implemented in the Seurat framework, and there are various ways in which you can tweak this. Thresholds you can set, you can change the genes that you use for this, you can use different methods for deciding on how you pick the nearest neighbors to each of your cells. There's a number of different things that you can adjust with that. But because more and more people are using this, Seurat have sort of streamlined this integration process, where now the integration is done with a very small amount of very simple code. So this is from their vignette for doing data integration, and they have a single integrate layers function where you pass in your Seurat object, you say which version of the integration you want to use. So there are different methods that you can use for finding the nearest neighbors, and what you want to call the newly integrated data. And that's great because it's really simple and it's really easy to try out. But it's also a very black box. Okay, you say integrate your data and you don't get much sort of insight into exactly what it has done and the connections that it's made. So that's where we're going to focus on today. So not changing our ability to do integration or even the method of integration, but trying to get some insight into how exactly that integration has been performed. So we're going to look at a visualization, and the visualization is going to mimic the slide that I showed you, in that it's going to try and allow us to see the the anchors that were defined when we integrated the data and find a sensible visualization of those. So whether or not we choose to go ahead with the integration, we can at least see how it's going to be structured, and we may be able to use that also just to visualize this on our data without performing integration. The software that we're going to use for this is an R package called Anchorview. It's hosted on GitHub. The address is up here, and it sits on top of Seurat and provides some additional functions which work alongside the core sort of Seurat functions to give you some new visualizations and some new methods that help with this. It's not installed on CRAN, so the installation of this just happens from GitHub directly, so you can do this with either the remotes package or with DevTools, both of which allow you to directly install from GitHub. So super simple to do, and will get you the additional package that you can then try out and look at the visualization yourself. So let's switch across. I'm going to move across to RStudio and actually do a walkthrough of some of the main features in the Anchorview package and show you how it works on some real data. Let's switch across to my RStudio. Okay, so my RStudio is here. And I've got an R notebook open, and I've done a little bit of work in here already, just to save some time. So I've just loaded in the packages that we're going to use from here. So I've loaded in Seurat, because we're building on top of Seurat. I've loaded in Anchorview, and I've also just loaded in the generic tidyverse packages as for general data manipulation. I've loaded in a data set that I've previously done some processing on. So the data that we have down here is just standard Seurat object. We have a look at this. I've done some processing this already, so it's a standard RNA data set. I've done the data normalization and scaling. I've done the dimensionality reduction already, so we've got both a PCA and a UMAP reduction in here, and we also have clusters that are called on this, just to save time in the demonstration. But all of that is completely standard sort of Seurat stuff. Let's have a look at the UMAP spread for this data. We have two samples in here, and those samples are distinguished by a metadata property called stim for stimulated. So I can do a dim plot of this data, and I will group by stim, just to have a look. Okay, and you can see that this is a data set which has the problem that we talked about. So we have two samples, the control and the stimulated, and they really don't intersect in any meaningful way. But just looking at this, you can get an idea of probably which cell types relate to which cell types there. If I plot this out, but instead of highlighting the different samples in here, I just do a standard dim plot and highlight the clusters. So normally the process for doing this is you do clusters on your data and then analyze the clusters. So if I just do a standard dim plot of my data, which should then highlight the clusters, we can see that the data clusters, okay, we can do them there. But what we'll find is that the clusters that are built from here are pretty much going to be composed of cells from one of our samples or the other. There's going to be relatively little overlap. And we can sort of formalize this a little more. It's quite sort of instructive to do something like this. So if I take the data on here and I just extract out the metadata from it. So we have the stimulated values in here, and then we've got the Seurat clusters down here. I can transform this just into a table. We'll make that into something that's got a cell ID from there. We'll group by our stimulus values and the Seurat clusters, and we can count what's in there. So we can see for each cluster the number of either control or stimulated cells that we have within that cluster. We can then, let's do a little visualization of this. We need to spread that out a bit. So we'll make this N. So we can now get separate columns for the control and stimulated count for each of the clusters. And we can already get a pretty good idea that each of our clusters is going to be dominated either by the stimulated or the control. This is the problem that we're going to have, that we don't get clusters that are nicely composed of cells from both of our original samples. They're all going to be one or the other because of the lack of overlap in the original dimensionality reduction. Now, to do the integration, we're going to use the sort of older style workflow for integration in Seurat. And what that's going to do is that we've at the moment got a single Seurat object that has the cells from both of our samples, and we're going to split that so that we will end up with separate Seurat objects for the control and the stimulated sample. So I can just use the method in Seurat called split object, and then we can say split by, and it's going to be by the stimulus column, okay, in our metadata. Okay, so that will separate these. So if I save that into split data, okay, and I have a look at split data, I've now got a list of two Seurat objects, one for the control and one for the stimulated. Okay, so those will be split out that way. I'm now going to find the integration anchors. Again, this is standard Seurat workflow. We haven't got into the Anchorview stuff yet. We're going to use the find integration anchors function from Seurat, where we take our split data and we can say which method we want to use to find our nearest neighbors. We can do these scRNAs, this sort of correlation analysis, or RPCA for reverse PCAs, beyond the scope of today, but this is one of the things that you can play around with and try it out. So I'll do the RPCA method for finding the integration anchors, mostly because it's quicker. But also works very nicely as well. So I will do that. This will take a little time to run, but it's now going to find the integration anchors between those two data sets and will give me back an integration anchor Seurat object in there. And from that point, we can start doing Anchorview things with that information. Should take maybe sort of 30 seconds or so to do on this. There we go. So we've now got a set of nearly 9,000 anchors across the data that we've got. I want to annotate those anchors now. So at the moment, the anchors are, let's see what we see in here. So these are just an anchor set object between two Seurat objects in here. And it says that you can plug this into the integrate data function, so that would actually perform the integration, but we're not going to go that far. We're just going to look at the properties of the anchors in here. The first thing I want to do from Anchorview is to annotate those anchors with the positions of the ends of those anchors inside our dimensionality reduction. So in this case, inside the UMAP that we have across the data. So I'm going to run the first Anchorview function, which is get positions for anchors, where we can just put the anchors that we've just put in, and we need to say which reduction we want to use, which dimensionality reduction do we want to use, and in our case, we want to use the UMAP positions. And I'll save that into anchor positions. And if we have a quick look, we can see what we're getting now are the cells from the two data sets. So cell ID 1 is the cell in dataset 1, cell ID 2 is the cell in dataset 2, and then we have the positions of those cells on the X and the Y in the UMAP dimensionality reduction that we have from there. From that data, we can now plot out our first view. Now, this is going to be a little messy because we haven't cut these anchors down in any way. We have a lot of anchors at this point in the process. But let's do this. I'm going to make this a little bit bigger as well. And we will plot the integration anchors. So we need to give this 2 bits of data. We need to give it the split data. So that's the list of the 2 Seurat objects that we had. And we need to give it the anchor positions that we just calculated. And we need to say which reduction we're using for this. So it must be the same one that we used when we calculated the positions. And that will get us our first meaningful plot on here, albeit that it's going to be a little messy. So it's going to take a little time because it's going to have a lot of cells in here and a lot of connections for the anchors, but we'll see ways in which we can make this a bit nicer in a second. But let's get the full data set first. Poor little computer. We'll get there in a second. It's longer and you have more people waiting for it. I know you work. I did you earlier on today. Hey, there we go. Told you it was going to be messy. So what we've got is the UMAP spread between the 2 data sets and all of the anchors overlaid. And you can see there are a lot of them. Okay, so it's managed to find a lot of mutual nearest neighbors in here. Now, this is messy, even though we can kind of see some of the structure that we have in here. Now, we can make this cleaner in a very crude way. So if I just copy this, I could make this cleaner by just randomly sampling the anchors that we put in here. So instead of plotting all of the anchor positions in here, I could say, let's take the anchor positions and do sample N500. Okay, so we can just randomly pick 500 anchors from the set and plot that, instead of the 8,000 that we had before. That'll come up a lot quicker, and we can see the structure a little more. Now, this isn't great in that we're just randomly sampling anchors from here, but it does mean that we can see the structure a little more clearly, and we can see which cells are being connected to which. Rather than doing that, what you can also see in here is that there are plenty of instances where you have anchors that are very close to each other at both ends of the anchor connection. So rather than plotting every individual anchor or just randomly sampling, we can cluster the anchors that are in there. So if we have a set of anchors that all start in a very similar XY position and all finish in a very similar XY position, we can merge them together. So this will be the next sort of stage of cleanup that we can do in here. So again, from within the package, we have this thing that says cluster anchor positions. I'm going to take the anchor positions that we calculated before, and then we can just say what the cluster threshold is, which is basically how far apart can the endpoints be and still be allowed to cluster. Okay, so you can play around with this to see what you've got. So I will do that, save that into clustered anchors, which will take a second to calculate. And now it's going to combine the, what was it, 8,000 odd anchors that we originally had down into a much smaller number of those. And let's have a look at what's in these. So if I take these and with every anchor, we will have what's called an anchor weight, which basically says how many anchors were combined together to in the clustering. And let's just look at the first few of those. So here we have the same XY positions as before, but at the end we have the anchor weights. So in this case, you can see that we have like nearly 700 individual anchors that were all so close to each other that they've been grouped together. Okay, and at the top, that will be obviously very high. At the bottom, we will get ones that are much more lowly weighted, where it really was just a single one and none of the surrounding cells supported it. Okay, so we can use this. We can now redo our plot, but now when we're plotting, we'll use the clustered anchors instead of the original ones. So let me just copy this because it's basically the same plot as this. So it's the same plot integration anchors, but now instead of the anchor positions, we're using clustered anchors. And we should see that it's a lot quicker to draw, but also that it's a lot less messy because now we have the thickness of the line relating to how many anchors were combined from those points. So we can see some nice thick lines that represent the main sort of groups of cells that have been put together, but then supplemented with a few sort of spidery ones, which may well be mispredictions. If you look over here, for example, there's a nice strong line going down to there, but there's a little spidery one that goes over here. So we can clean this up even further if we filter our clustered anchors based on their weights. To say, well, just only show me the ones where there was actually support from many anchors in the same place, which should then sort of clear this up a bit. So if instead of putting our full clustered anchors in, we can say, let's take our cluster anchors and filter for the anchor weight being greater than 50, for example. And if we do that, we should get a much cleaner view. So now we really have got quite a nice view of which parts of our UMAP Seurat would want to align together, and we can pick them out from this. So we can see these structures sort of mirror across here. This one is going down to here. This is over here. And basically what you would expect from this data from a visual inspection. This one is not too hard to do, but in other cases it may be a little more challenging. Now, we are connecting at individual cell levels in here, and when we're clustering, we're just clustering arbitrary cells together. But another way which is really quite useful to think about the integration of data is to run the clustering of data individually for our split data set. So we have our control and our stimulated. So we can define clusters in both of those individually. Okay? And then we can start looking for connections between clusters in the different data sets. So let's start by just redoing our clustering. So the clustering we haven't run yet. It just came in the data that I'd loaded. But in my split data, I'm going to do separately for the two subsets of this, the control and the stimulated, and I'm just going to run the standard clustering. I'm not going to try and optimize this or anything. I'm just going to find neighbors in it and then find clusters. So again, standard Seurat workflow. Let's say resolution is 0.2, and I'll overwrite the first one. So this is just taking the first sample, which I think was the control one, recalling clusters in it, and then overwriting that, which will take a second. Okay, and then I'll do exactly the same thing for the second one in here. So now we have newly defined clusters in both of those. We can have a look at the dimensionality reduction plots in the two data sets with the clusters overlaid on them. I'll put these side by side. So let's say width is 12, 5, because we're going to put them next to each other. I'm going to use a function out of the grid extra package, nothing to do with the dimensionality reduction. It just lets me put two plots side by side. Grid arrange, and I'm going to arrange the two dim plots. So I'll do a dim plot of the split data 1, and I will do equals true, just so we can see where the which the clusters are, and we can do a dim plot of split data 2. So we're just doing standard dimensionality reduction plots, just standard UMAP plots, but hopefully they will go now next to each other. So here we have the clusters called on our two datas, the control and the stimulated, and you can see again they don't sit on top of each other, and now we have clusters called separately in each of these. So we can start making connections now between the clusters in this data set and the clusters in this data set, because it's hopeful or likely that there will be an equivalent cluster that we find in each of the two data sets, and we can connect at that level. So again, we need to run our integration anchor finding because we've messed around with our data. So I'm now going to find my integration anchors again. Integration anchors, this is going to be on my newly modified split data. I'll do the RPCA again, and I'll call these root. Okay, so we're just going to rerun the integration anchoring on the newly clustered individual samples. And then we're going to do some cool things with this. There we go. So finds a set of anchors again. And now we have some other functions that we can use from Anchorview to be able to look at this. So the basic one, which is maybe the most useful in this, is to plot the cluster interaction matrix. So this is going to give us a heat map of all the clusters in one data set against all the clusters in the other, and showing us which combinations of cells are most commonly joined between those. So do this with the grouped anchors, and that gives us a really clear view. Okay, so on our original data, we can see that cluster 5 was cluster 5, cluster 0 in the first one was cluster 1 in there, cluster 8 was cluster 7. There's a little bit of sort of off target in here, but mostly we can see there's a really nice one-to-one correspondence between the two. Now, this is just collecting clusters that we've defined in here, but actually on this data set, one of our samples has actually got cell types annotated into it. So we don't have to plot between sort of Seurat clusters each time. If we have cell type annotations, we can use this. And this is a really nice way of being able to transfer cell type annotations over to a new data set that you haven't yet labeled. So if I run this same code again, of my grouped anchors, but this time I can say that my clusters 1, so which metadata it's going to take for the clusters in dataset 1, is called Seurat annotations. Then instead of just using the Seurat clusters numbered, it'll actually put the named annotation in there. So here we go. So now we've got the different cell types that were annotated onto one of our data sets, and we can match that up against the clusters that we defined in the other one. And sometimes we get really nice single matches. So MK is cluster 8, activated T cells, cluster 5, CD16 monocytes, cluster 4. Sometimes it's a little bit more fuzzy. So yeah, maybe the B cells and the B cells activated are both in cluster 2. So that could maybe help us define and go, oh, maybe I need to increase my resolution a bit. And I can do that. Likewise, the CD4s are both falling into cluster 0, and maybe we can separate those out. But where you have this kind of annotation available, you don't necessarily need to do the full integration. You can just run the integration anchoring part and then can use this kind of visualization to work out which of the clusters in your own data represent which cell types. The final plot that I want to put out is we can do a UMAP plot linking the matched clusters, the two. So we've done the matching in just a matrix here, but the final one that I'll do is to do a dimensionality reduction plot for our clustered interactions. So we're going to do this with the grouped anchors. And we'll say that the clusters that we want to do is going to be just the standard. Clusters 2 is also that. Our reduction is going to be UMAP. And we can also filter a bit. So again, we can say what percentage of cells need to be connected between the clusters to draw the actual connection on here. We can see that it's a pretty nice match in, well, certainly in the original data, we get a very complete match in here. But if I don't put a little bit of filtering, then some of these slight off targets will also get drawn in as well. So here, if I say min percent equals 30, then it'll only draw me linkages that are more than 30% of the cells between those clusters. And that should give me, there we go, a final view where we can see the links between the clusters that are on here. So now we have a really clear view of this. And we could either use that to refine our anchoring strategy or refine our clustering, or just use it as a visualization for the different types of data that we have. And that's it. So hopefully that's given you a feel for what you can actually do with this package and how you might find that useful in understanding the structure in your own data, sorting out the annotations in your data, or just making the actual integration process less of a black box when you come to do it.